we come to the next presentation that will be given by Dr. Prabitha Urwyler. She is going to talk about polymeric cantilever arrays that are used for biomedical applications, which includes the detecting of DNA fragments and of metal ions that are relevant. Okay, Prabitha, the floor is yours.
Yeah, thank you, Bert. Thank you, Bert, for the introduction. So good afternoon, everybody. So I'll be away from cells, so you'll have to excuse me. This is my PhD work from 2008 to 2011, and I'm currently a postdoc at University of Berne, but my work will be with Professor Bert Müller, the work I'm presenting. I'm currently at the University of Berne. So what I'm going to talk to you is how do we apply polymeric microcantilever arrays to detect DNA fragments and metallic ions, which have physiological importance. And to start with, the motivation is to apply cantilevers in biosensing. So what is biosensing? So biosensing is detection of specific analyte binding, sample analyte binding via a transduction. And depending on the type of the transduction signal, there are different types of transducers. So there are optical transducers, electrical transducers, mechanical transducers. So cantilevers belong to the mechanical transducers. And why are cantilevers important, or why do we need to get into this field? Is because with cantilevers we don't need the tagging, the fluorescence tagging, no labeling required, rapid detection, and cantilevers are small in size, so the volume required, so the sample size is very low. And commercially we have silicon microcantilevers which have been in the market and are being used. And so basically coming to the theory behind cantilevers. So what are cantilevers? So cantilevers are basically mechanical elements which are fixed at one end and are free to bend on the other end. Sorry, I go back. So the cantilevers, the mechanical properties of cantilevers are governed by the element, the mechanical properties of the cantilever element, the Young's modulus ratio, and the geometry of the cantilevers, which then govern the spring constant and the resonance frequency of the cantilevers. Because cantilevers in biosensing are operated in two modes, that's the static mode and the dynamic mode. In the static mode, the bending is governed with a spring constant, and that's where we measure a static bending. Whereas in resonance, in the dynamic mode, the shift in the resonance frequency is measured, and this is it tells you how much of mass is attached or adsorbed to the surface of the cantilever. And in addition, in our study, we concentrated only onto the static mode, and we took into consideration the mechanical bending of the cantilever. And as you saw in the small picture on the left-hand side, as this analytes bend onto the functionalized surface of the cantilever, there's a differential stress on two sides of the cantilever, and this is governed by the Stoney formula, which has been done quite early in physics. And in our static mode of bending, what is very important is the spring constant, the factor K. And the factor K is proportional directly to T, that's the thickness of the cantilever, and L was the length of the cantilever. While we in our project have developed the cantilevers for a predefined size because we were trying to replace silicon cantilever, so the length was defined. We only had to play with the thickness of the cantilever. So that was again governed by the Young's modulus of the polymeric material we were using. So as I said, we were fabricating cantilevers which were in dimensions exactly to the commercial available silicon cantilevers. So the spin-off of Universal Cantisense Research System has a product which they also they use commercially available silicon cantilevers, and they do have bioanalytic experiments using silicon cantilevers. So what you see there is a cantilever array with 8 cantilevers, and these are the dimensions we were supposed to produce, and to replace the silicon cantilevers with polymeric cantilevers. And the Cantisense Research System also uses the optical detection system to measure the bending of the cantilever. So the goal of our study was to mass produce polymeric microcantilever arrays which were comparable in size and signal sensitivity silicon microcantilevers, which were low cost, disposable, and which could be used for sensing experiments. So to begin with, we had to fabricate the mold, then injection mold these cantilevers, characterize the cantilevers for its mechanical properties, deflection, and then go on to sensing experiments. So I start with the injection molding results. So injection molding, as you see, we need to do quite some iterations. So first you have a steel mold where we laser ablated it, and the first generation of cantilevers had a thickness of 50 micrometers, and 50 micrometers was not enough for deflection. It was too thick. And so we went on to a second generation of microcantilevers where we arrived at 35 micrometer thickness. And you see the dimensions of the cantilevers are 500 micrometer long, 100 micrometer broad in width, and 35 micrometer in thickness. So the whole idea behind what I am showing you this figure is the theory behind filling a cavity in injection molding. So the outer wall, the mold, steel, is maintained at a temperature which is usually the transition temperature of the material, and there's a liquid polymer melt coming at the melting temperature, and the mold is cold and the polymer is hot. So as soon as the outermost polymers reach the cold mold wall, they freeze. So there is a difference in orientation of the polymeric structures at the end of the mold wall and at the center of the cantilever, and this also affects the mechanical properties of the cantilever. So the bending is different because the orientation of the polymer molecules are different. So we had to then move on due to the thick cantilevers. We then moved on to get rid of this cold wall immediate freezing to what is called variothermal injection molding. So here the injection molding cycle is longer, so the polymers we could achieve thinner polymers, 25 micrometer. We had a dedicated mold. We had heating on both sides of the cantilever of the mold, steel molds, and we achieved complete filling. As you see, full cantilevers with the air duct also filled. And with these cantilevers, we moved on to the characterization and the biosensing experiments. So we characterized them with the Swiss light source. So what we deduce out of the characterization is the central region of the cantilevers is homogeneous for all materials that we produced. We produced cantilevers in 4 different polymers: polypropylene, polyvinylidene fluoride, polypom, as well as PEEK. But unfortunately, PEEK could not be filled at full cavities because of its high elevated thermal properties. And I'll move on to the characterization. So for the characterization, what do? You have your cantilever. You have to coat them with gold on one side, basically for 2 reasons. Gold, due to SAM layers, it's very important to have clean gold surfaces, as well as for optical refraction, because we need to read the laser reflected right, and that's why we needed the gold on one side of the cantilever. So another reason is to also measure the thermomechanic activity of the cantilever to see if the bending is even. So we heat the cantilever from here. On the left-hand side, you see the deflection of the cantilevers when it's heated. So you need to know that all 8 cantilevers are bending equally, or you need to have a baseline, and these are the controls which we do. And on the right-hand side, what you see is what happens to a cantilever when there is surface stress generated. So we test that by forming a SAM using thiols, and you can see here the 8 cantilevers are 8. We have just 7 here. Is there a pointer here? Okay, yeah. So this is what you see here, how the cantilever bends when surface molecules are attached. So the cantilever bends down, and this is what you measure here. So this was a proof of control to show that the cantilevers that we have produced bend, and we could see it's mechanically viable. So once that was done, we moved on to sense copper ions. So the idea behind this was to measure transition and heavy metals in human blood samples or urine samples. And the binder for copper ions, what we took here was the glutathione. It's a tripeptide, and it has 2 carboxyl groups which attach the carbon ions, divalent ions. And this is what we wanted to measure. And the glutathione forms SAMs. This is self-assembled monolayers on gold, because we need that as a functionalization layer on the cantilever so that the copper can then attach once we have it. So how do we do the functionalization? As we have a chamber from cantilever, from the CantiSense, and we have 8 cantilevers basically in our cantilever array, and we just functionalize 2, 4, 6, and 8 with GHS, and the other 4 with a reference. So there should not have been a binding on the other 4. And even if there was a binding from other things, then we measure the deflection, the differential deflection. So what you see here is when we injected copper, the initial, this is due to the valve systems, we get some deviation in the whole system. And then you see here we have the colors mentioned, and here we have the differential deflection. So for 100 nanometer copper ions, we got a signal of around 36 plus or minus 5 nanometer. So this is just a motivation to to show you that we could use mass fabricated polymeric microcantilevers for detecting other types of metal ions in human blood samples. And the next one is we showed about DNA hybridization. This has been done in silicon, and we just wanted to replicate this experiment using polymeric microcantilevers. So basically the idea is we had to show this, the hybridization, that is the 2 complementary strands of of the DNA. And we had 2 DNA probes. One is the SF162, and the other one was the NL40. These are HIV strands. And then we had this sample DNA, which was complementary to the red one, that is the SF162. So out of the 8 cantilever fingers, we had all the even numbers. We had 1 of them coated functionalized with SF, and 4 of them functionalized with SF, and 4 functionalized with NL. So, and then we had a sample that is 100 microliter of 1 micromolar SF162 into our sample, and we measured the differential signal between the 2 different cantilever arms. So basically, our sample DNA should have just binded to its complement, and we measured the bending, which was the complementary interaction. So this is the results. Basically, what you see here, we have the baseline here, and we injected. So here we had 4 cantilevers functionalized with the red DNA, and 4 cantilevers functionalized with the blue DNA, that's the NL. So then we had injected the complementary SF162, the green one. So when the green complemented with its complementary, we saw a deflection of around 7 nanometers. And just to make sure, as the earlier speaker said about controls, we did a control. So we injected few more of our samples to see if everything was saturated. And so some of them which were not binded, and some in between which were left out, we could see a small signal of 2 nanometers. We then repeated this experiment by flushing out with urea, and in the end we could show this signal, which for the experiments that with another experiment we got a final signal of 7 nanometers for this hybridization of DNA, and this was a test for our specificity, and we were close to what silicon has produced. And with this, I would like to conclude that with injection molding, we can fabricate microcantilevers, which can be used for sensing medically relevant species, and yeah, there is scope for improvement here. Thank you.
Oh, thank you very much, Prabitha. I think there are some questions from the audience. Yes, please.
Thank you for your presentation. I guess the first report regarding this microcantilever has been almost 2 decades, I think.
Yes.
And still, I get similar mechanism and picture in this field. I'm not doing this field. So how do you expect the most peculiar advantage of using this cantilever as sensors, or the market size or something like that? Can you comment on that?
Yes, I do agree with you that it's been here since 2 decades. The one that has been used is only the silicon microcantilevers, and the whole area of silicon microcantilevers, they are more than 100 Swiss francs. And if you want to use it next to the lab site, we are trying to aim at low cost and disposable use and throw, and that is why we wanted to achieve the whole thing, what we could achieve with silicon microcantilevers using polymeric, and that is why we moved on. And another advantage here is, if you look at here, in addition, during injection molding, we wanted to pattern these microcantilevers with different patterns, and then we wanted to grow cells on them and to measure cell bending, so we could go into another field of microcantilevers.
So is it currently, used in clinical application or something?
The silicon microcantilevers, yes. They are at University Basel, they are using it, but it's not clinic. I mean, I haven't seen anything out. But yes, Professor Müller is here now, currently going forward in this direction.